Welcome to today's division. So we are going to continue our division for biology. So in today's class, we are going to be revising biology 2023. So don't forget to subscribe so that you don't miss out the next questions when they'll be posted. And don't forget to also join our first revision academy. So the number is on the description. And also we have a special class for free for those that want extensive revision. So to begin, the question is saying figure 1 shows changes which take place when a plant cell is placed in a solution of known concentration from stage 1 up to stage 3. So they are saying that this diagram here represents the changes which take place in a plant cell when it is placed in a certain solution. So now our first question is, identify the parts of the cell labeled A and B. So what is the first thing you need to know when you are answering question in biology? Because you don't upload a question in biology anyhow. So the first step, you need to identify which topic is this question coming from. That's when now you understand that, okay, the points I should write are this and this. So this question is coming from the first topic in biology, which is cells and cell organization. This is a topic where we look at osmosis, diffusion, and active transport. So here we can say that this question is coming from what? Osmosis. Because on osmosis, that's where now we look at different type of solution with different concentration. Remember I told you we have hypertonic, hypotonic, and isotonic. These are some of the solution. So if you want to learn more about it, I have posted a video on osmosis where I've explained all the three terms. The link will be put in the description. Go and watch it. So how can we identify now our cell? So of course, this is a simple diagram of a simple cell. We know that the first part is always the cell wall. Then it is followed by what? The cell membrane. Then followed by A, which is the cytoplasm. Then we have this part which is known as the nucleus. Then part B is known as the vacuum. So don't get yourself confused with the vacuum and the nucleus. These are two different things. Then here now you can write our A is the cytoplasm. Then our B is the vacuum. Yeah. So moving on to our second question, which is saying, using the changes shown in figure 1, explain what could have been the concentration of the solution in which the cell was placed compared to the cell sap. So here they want us now to explain what really happened for the cell to move from this state to this and finally this. We are seeing that here the cell is what? Is slowly shrinking. So what could be the reason why the cell is shrinking? So how can you explain it? So you can explain it in the sense that this cell was, so the cell was placed in an hypertonic solution. So for those of you who don't understand the meaning of hypertonic solution, we are saying that when something is hypertonic, it means it has a lesser concentration. What do we mean? So something which is hypertonic, remember guys, it has what? Less concentration, or it has a low concentration. In short, I'm sorry, it has a higher concentration in short. So when we're talking about something which is hypertonic, it has a higher what? Concentration. So meaning if something has a higher concentration, meaning it has low water. So meaning water will start moving from the cell outside. So when water start moving from inside the cell, going outside the cell, it will causes the cell now to start what? Shrinking, which can lead to how the cell is looking on stage 3. So now here you can say the cell was placed in an hypertonic solution and it lost water by osmosis. Full stop. So this you have explained. Why? Using the changes, explain what could have been the concentration of the solution in which the cell was placed compared to the cell sum. So the cell was placed in an hypertonic solution and it lost water by osmosis. Why? Because the solution had a higher concentration than the cell. Yeah. So meaning if the concentration outside the cell is high, the cell will start losing water so that the concentrated part also become what? Diluted. So this is what you need to know on hypertonic. Then when you talk about hypotonic, in short, this is a solution which has a lower concentration. Therefore, if a cell is placed in a solution with a what? A lower concentration than the cell inside, it will do what? It will start gaining water. You have seen that? So this is what you need to understand. Then the next question is, what term is given to the process that lead to what? To changes shown in this figure. So I've already talked about it a lot of times. So this process, the entire process you are seeing here, is known as osmosis. So osmosis, this is the process where water molecule move from a place of higher water potential to the place with what? Lower water potential. We said that when something has a higher concentration, meaning it has low water potential. So meaning water will go to it. Then when something has a lower concentration, it means it has a higher water potential, meaning water will move away from it. That's what you need to understand. Then question C is saying, what would be the overall effect of the process on the plant? So the overall effect of this plant, which is placed in an hypertonic solution, is that the plant will lose its water and it will become what? Flaccid or flabby. What do we mean when you say flaccid? So it will become smaller and weaker. So that's flaccid. So the cell will become what? Flaccid meaning small or small and what? Weak. And also the plant will do what? Will start what? Wheating. So what is wheating? So wheating, this is the process where the plant starts sagging of what? Delicate plant parts such as the leaf. So you start seeing that the leaves will start to die slowly because they are losing water. That's the meaning of sagging. They will start to die slowly by losing water. So that's known as wheating. So the overall effect of osmosis, or the overall effect of placing a plant in an hypertonic solution, it is that it will cause the plant to become flaccid and wet. Yeah, so these are the two effects which you need to understand. Then the other question is saying, which of the three stages of the cell in figure 1 could be classified as turgid? So turgid state, this is a state which is, you can understand that the condition where the protoplasm of a plant places against the cell wall. So whenever you hear the word turgid, just remember that the protoplasm is placing against the cell wall. What is the protoplasm? So remember I told you that the protoplasm, contains three things. We have the nucleus, the cytoplasm, and the cell membrane. So when we say that the protoplasm is placing against the cell, we are saying that the cell membrane is attached to the cell wall. So among the three stages, which stage do you see the cell wall being attached to what? To the cell membrane being attached to the what? To the cell wall. So in short, it's stage 1, because stage 2, we see that the cell wall starts to what? To move away from, the cell membrane starts to move away from the cell wall. Again, here you can see that it has moved now even further away from what? From the cell wall. So meaning the stage which the plant is turgid is stage 1. So in biology, you have to understand the term before you answer, because most of you could have rushed to answer in what? Stage 3. So stage 3, the process where the plant has what? Has become small, is known as a plasmolyzed plant. So this is a plasmolyzed state. So what do we mean when we say a plasmolyzed state? So this is where now the protoplasm pulls away from what? From the cell wall. So whenever the protoplasm pulls away from the cell wall, it is known as plasmolyzed state. When the cell wall places against when the cell membrane places against the cell wall, it is known as turgid state. So these are the two things which you need to understand. Now, how does turgidity comes about? So turgidity, this comes about whenever you place something in a what? Hypotonic solution. So when something is placed in an hypertonic solution, the cell becomes what? Plasmolyzed. Yes. Then when you place something in an hypotonic solution to become turgid. So this is as a result of hypotonic. So these are the two things which you need to understand on osmosis. So in other way, they can change the question and bring what? Hypotonic. So you need to understand. Then the final question is saying, name two other processes which can make substances to move into and out of the cell. So the other processes which can cause the movement of substances in and out of the cell are active transport and diffusion. So everyone knows that active transport, this is the process by which ions are taken up by the plant. And diffusion, we know that this is a process by which substances diffuse from a area of higher concentration to an area of lower concentration. Here I like using an example of perfume. When something is wearing the perfume, once it enters into the room, that perfume will start diffusing, going to places with a lower concentration so that it can balance it up, which is known as equilibrium. So this is where our first question ends. Sticks through as we solve question 2.